നേരെ വേണമെങ്കിൽ സാറിനോട് ഇത് ചെയ്യാനും പറഞ്ഞു കേട്ടോ. അതായത് സ്ക്രീൻ ഷെയർ ചെയ്യാനുള്ള കാര്യങ്ങളൊക്കെ നോക്കാൻ പറഞ്ഞു കേട്ടോ. So what is the left meeting? 51. 3 minutes to go.
A warm welcome to the one and all gathered here. I, Neetu Ashok, cordially welcome you all to the International Conference on Ocean and Climate Change 2022, organized by IFCO Foundation's Kerala chapter. A small cheer and great welcome makes a merry feast. I sincerely welcome all those who believe in the power of nature and who would like to join me in our exploration journey. Today, here we are honored with the presence of two eminent personalities, Dr. Alan D. Wanamaker, the professor, Iowa State University, United States of America, and Dr. S. Bijoyanandan, the professor, Cochin University of Science and Technology, India. Our first session begins with the experienced words of Dr. S. Bijoyanandan on the topic Dimensions on Molluscan Diversity and Ecosystem Services in Planetary Boundary Framework. I repeat the topic Dimensions on Molluscan Diversity and Ecosystem Services in Planetary Boundary Framework. Dr. S. Bijoyanandan, the Professor, Department of Marine Biology, CUSAT, he has 27 years of research, development, and teaching experience in marine biology, climate science, invertebrate zoology, marine ecotoxicology. He has achieved 18 major research and consultancy projects, including UN Compensation Project in Arabian Gulf Coast, currently executing 11 projects as PI, also in Indian Arctic Research Project. He is the author and co-author to 220 journal articles, books and proceedings, edited 10 special volumes, recipient of Jawaharlal Nehru Award in 1993 of Indian Council of Agricultural Research for highly quality doctoral thesis in India, and UNESCO Fellowship in 2008. US Fulbright Visiting Scholar, Technical Member, Kerala State Pollution Control Board Fellow, and the coordinator of UGC SAP program of the Department of Marine Ecology. 26 students awarded PhD under his mentorship in CUSAT and other universities. His researches mainly focus on invertebrate zoology, marine biology, marine pollution, wetland ecology. Welcome, sir. Dear participants, to keep the quality of sound functional during the meeting, please mute yourself when you are not sharing. I may also mute the room if necessary, but please remember to unmute when you wish to share. Now, without any further ado, we'll turn the time over to Dr. S. Bijoy, sir.
Thank you. Thank you, Neetu. Okay. Good evening from India. Good evening to my friends associated with Accor Foundation and other institutions who have gathered here for this webinar of the International Webinar on Ocean and Climate Change. All of you can hear me? Neetu, it's audible for you?
Yes, sir, it's audible.
Okay, great. Thank you. I'm so happy to be part of this Accor Foundation International Webinar on Ocean and Climate Change. And at the outset, let me just thank Accor Foundation because they have appropriately and wisely selected this international webinar, especially titled Ocean and Climate. I don't know whether it's because we need to really put our soul and head, our heart and soul, in fact, into this looming threat of ocean changes because of climate, ocean change or ocean transformation because of climate change among what we all live in the coastal systems. And the coastal ecosystem largely associated with the marginal ocean systems. Globally, the climate is changing. Ocean has an important role. And today, with these two important lectures today, let us begin with the lecture. I will just now I'll share my PowerPoint, after which I will come to my discussion on various aspects. I think my lecture is scheduled for about almost half an hour, or maybe about 35 minutes. I hope to precisely keep my time. I think my PowerPoint is visible, my friends.
Yes, sir.
Yes,
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 sir.
thank you.
Yeah.
So today we start with discussion on the elaborate. It's a very wide topic, dimensions on molluscan diversity and ecosystem services in the planetary framework. In fact, when I was speaking to Jishnu, in fact, this is a very elaborate topic. It, in fact, requires hours to go through an entire aspect of this molluscan diversity, because because mollusca itself, as we all know, it's over 100,000 species, extent of species, and what is happening in terms of climate change is very complex. Still, many of the studies are still to ascertain seriously that the changes in Mytilus or changes in abalone or changes in clams is really substantiated. But to lead my colleagues in this forum to the different aspects of climate, let us just get through the basics. I think I'm not going to details. We'll just come to the real fact. But we have had 5 mass extinctions. When you quite compare with the one happening now, this is possibly the 6th, we believe, to be. This is rather concentrated on. We focus on caused by single species, or rather on a single species culture, because we have several invasive species, we have extinction of species, we have been through several mass extinctions, which I have depicted here. Of course, we are going through the Holocene extinction over 11,700 years ago to present. Of course, the mass extinction has a really a great influence, or a significant influence, on the molluscan resources, because largely the paleology, the paleoclimatology is largely dwelled upon the molluscan resources. Because the second talk by Dr. Allen from US, I think he will, my friend will, he will take you through the different aspects of its monitoring, on the geochemistry, the relationship of sclerochronology. That's a very significant topic where you will be able to link up. But I will just throw some insights on how this diversity is changing with times. Mass extinction has happened over where 50 out of 97% of the species of the earth have wiped out. Almost we call it as the events of the mass extinctions, because, and mind you, my friends, these molluscan resources are paleoclimatically and paleogeologically are really very important. And they are very significant in and related to the mass extinction, because over the Holocene period. And last major marine extinction ever occurred in 252 million years ago, and the next maybe just decades away. Of course, we have over a species of many species of extinctions are happening, because the next maybe, and now it may be happening anytime. And according to new research on past extinctions, we have, which is found in the oceans, rapid absorption of carbon dioxide. We know that carbon dioxide is almost it's boomeranging, it's going up, and it's almost, we are 400 plus of carbon dioxide in the atmosphere, ppm, and this is going to really have a serious implication on ocean acidification. And mind you, my friends, ocean acidification can relate to the ocean, the calcification of shells, the agaronite production, the calcite production, which is very much linked to the larval production and the larval development of the mollusk. So this is something which is very tricky. So if you go through the chronology, the carbon dioxide linked to climate science or the climate change is significantly harming. So where in this wonderful chart which was put forth by Stefan et al. In 2015, the estimates of different control variables on the planetary boundaries. I'm not going to elaborate all the planetary boundaries, but climate change is one of the significant planetary boundaries of the biosphere integrity. When you discuss on the 7 planetary boundaries, which was elaborated by Stefan, important articles, the important scientists, the important the previous researchers who have worked on different aspects of climate and biodiversity, the warning on the significant extinction of species has been well postulated now. It is well established. Mass extinctions are related largely because of the ocean acidification, as far as oceans and the current increasing ocean acidity increasing faster and present. We know that over 6 billion fish, almost 6 billion tons of fish and invertebrates have been taken from the oceans since 1950. We have exploited. We have had over carbon dioxide concentration is at least 2 million, highest in the last 2 million years. Almost, I would say, it's the most warmest period in 2020 to 2021 period. Sea level rise is the fastest over the last 3,000 years. Of course, having a serious influence on the molluscan and other resources of the. Similarly, Arctic sea ice, lowest. Similarly, the extreme heat, ocean warming, freshwater habitats, and the life source of life are all human. Yet they are all most threatened, strongly affected. If you look at the plastic pollution as a major source of, and also including we are collecting plastic materials or the microplastics even from the Mariana Trench. Human activity is threatening, and these planetary boundaries are they significantly influencing? We are crossing the red boundaries under the planetary designations put forth. If you have almost the coastal development, the population and growth, pollution, and 50% of the salt marshes, 35% of the mangroves are lost. We have almost lost 30% of the coral reefs. 29% of the seagrasses are almost lost or possibly degraded worldwide. So this impact is definitely 89% of the oyster reefs may have been lost globally. We have wonderful coral reefs globally. Overfishing has been persistent, I'm telling you, including marine mollusks. Marine environments are lost fisheries and linked to declining water quality, increasing occurrence of harmful algal blooms, offshore pollution, and oxygen depletion, hypoxia. All these are exacerbating to the larger phenomenon of climate change linked to the planetary boundaries. If you look into the human activities which has threatened the ecosystem services and the coastal marine biodiversity, the ecosystems provide a range of services that are fundamental importance to human survival and their protection and livelihood and also their health. So the report by Barber in 2017, if you look the seaward edge in the inland boundaries, the mangrove scope, which is put across in the current biology, you can see the marine ecosystem providing the ecosystem services and the ecosystems, economic ecosystem. So the well-being are all connected. So the functions, the structure, the goods and services are all connected to the economic system. So if you look at the multiple scales of climate regulation and carbon sequestration, global scale to storm protection, to recreation, to food supply at local and regional scales, estimate that the economic valuation, the economic system which provides in since 2011 is almost 145 trillion US dollars per year. This is as per the Costanza 2014 as well, the global values in the coastal and marine ecosystems contribute significantly to this. And this significantly is structuring our biodiversity. Maybe largely from the Paleozoic period to the Carbonaceous period to the Permian period, Triassic period, and of course the Cenozoic era. So all through this 4,500 million years ago, when the Earth forms almost, and to the present period beyond the Holocene period, this change and how many species inhabit Earth, yes, the answer to the question remains still enigmatic as efforts to sample world's biodiversity to date has been limited. This has been a direct quantification of the global species richness, and because indirect effects rely on the assumptions that are proven highly controversial. Globally, if you estimate, you have about taxonomic experts estimate that the estimates of species may be about between 3 to 100 million species. And there may be, they have, these species are complementary. 4 complementary perspectives of biodiversity may be at the gene level, species level, ecosystem level, landscape level, all linking to the different services they provide, maybe to the nature conservation, to the provisioning services, to the cultural services and the regulatory services, all interlinked to the composition, stock, structure, biodiversity functions. So all these factors linking to the supply of ecosystem services, all are linked to the exemplary resulting ecosystem functions, maybe filtering, pollination, secondary production, groundwater storage, temperature, soil formation, carbon sequestration are all linked, all as part of the ecosystem services. But if you look at the life forms, where studies predict that over 8.7 million eukaryotic species globally, of which 2.2 million are marine, in spite of the 250 years of taxonomic classification and 1.2 million years of already cataloged central database, result in over 86% percentage of existing species on Earth, 91 species of ocean still await description. So we have hardly described over 8 to 9% of the ocean species. Many are endangered, many are threatened. And if you look at the molluscan species, over 90,000 to about 100,000 species are described. And mollusks are the second largest phylum which is described. If you look at the different Mesozoa, Hemichordata, the Ctenophora, Rotifera, Echinoderms, Amphibia, Insecta, if you see, and this is the second largest phylum which is described in the extant species, range from few millimeters to over 20 meters. And largely depicting in the ocean biogeographic information system. If you go to the biodiversity platform, sharing platform of Clearinghouse, one of the census of marine life, you can see the international project planning about 10 years that recorded diversity, distribution, and abundance of research, where you get information on the, other than including the molluscan species, more than 2,700 scientists from 80 nations, 540 marine expeditions, and reported from the Royal Society of London in October 2010, the Ocean Biogeographic Information System, and also the molluscan base, the worms. So all these research and the marine life put across our information on mollusk and associated species. So animals include 30 major phyla, of which 2 phylum, Arthropoda and Mollusca, dominate the diversity, the latter composing almost a quarter described in marine animals. If you look at the Wagner and Wolfson 1918, you can see the over the evolutionary growth during the Precambrian, Cambrian, Ordovician, Silurian, Devonian, Carbonaceous, the Permian, and the Triassic changes to the various forms of molluscan species. If you look, the giant clams and the smaller cockles use sunlight through photosynthetic associations with single-celled algae like corals, while several groups of bivalves gain nutrition from the symbiosis of chemoautotropic bacteria, some even losing their guts. Mollusks are integral to marine food webs. They can prey, they can be consumers, include suspension feeders, deposit feeders, herbivores, or predators or parasites. So mind you, friends, mollusks are really an integral part of our ecosystem, the marine ecosystem. The diversity is known largely, as I highlighted about 10 p. They appear from the Cambrian, and Mussel Watch program has been one of the leading species analysis program where it provides the changes in environment related to the molluscan resources, because it is used as a sentinel species to assess the state of your environment, especially in marine ecosystems. It is used in biomedical research. It's used in pest analysis. As a pest, it is invasive. It is also performs its role as an intermediate host in parasites. Mollusks are best known for the diverse phylum, as I said, over 85,000 extent to over 100,000 species, valid species in addition to about 3,000, almost 35,000 described fossil species by Zhang in 2015. I've just shown you Polyplectophora, Neomorpha, and all these different kinds of, as described by Vaniger and Olsen in 2015. Scaphopoda, Bivalvia, Gastropoda are all described. They're interesting from an evolutionary angle. If you look at the species richness, they are huge body with body plans of phenotypes is exhibited in its representatives. It includes maybe cylindrical in nature, shell-less, spicule forms, worm-like crawling, or maybe 8-shelled grazers, 2-valved filter feeders, as in the case of mussels and clams, single-celled, as in the case of Monoplectophora or Tribelidia, Gastropoda, that is in the case of snails and snugs, Scaphopoda, maybe tusk shells, Cephalopoda, as in the case of octopus, squids, and nautiluses. Similarly, the origin of this phylum dates back to early Cambrian. That's what I highlighted, almost 540 million years ago, but most likely to be deep-rooted in the Precambrian. It's in the Vendian era, as per the Prakev in 2008. So you see the diversity, the distribution pattern. Mollusk shells originated, actually they have a diverse morphological kit in hand, and mollusks are able to conquer both aquatic, terrestrial, and you can see from the mountain ranges, you can see in the terrestrial areas, you can see in the deep oceans, you can see in the coastal sections, in the littoral habitats, and they are largely. You can also see the spectacular flying squid, the Todarus pacificus, are even able to get partly airborne by using its fins and arms. You can see the flying squid here on the right side. Japanese researchers confirm squid as fly, jet launch, and glide them to the air square. Can fly almost 30 meters in 3 seconds. That is Toradus pacificus. It's got a length of about 203 to 225 millimeters. This is reported from the Hokkaido University. You can see its gliding nature. I have just depicted here with this. So also molluscan diversity in aspects of its biology, maybe in morphology, physiology, behavior, genetics, ecology, and additionally includes a long and extremely rich and informative fossil record. Most familiar groups are the limpets, whelks, maybe the snails, and also the snails and slugs, generally called as the gastropods. Over 70,000, almost 100,000 species. Clams, as you generally have mussels and scallops. Class, they belong to bivalves, lamellibranchia or pelecypoda. Over 20,000 species. Squids and octopus class cephalopoda, almost 900 extant species. Maybe 40,000 over fossil species are there. Molluscan species, if you look at over 100,000 species recorded from over the world, 80 to 100,000. 80,000 to 100,000 species recorded around the world. Over 3,270 species have been reported from India. As far as in the Indian subcontinent is concerned, it's got an abundant molluscan sources belonging to 220 families, 591 genera. Bivalves are one of the diverse groups, but facing severe case of climate extinction. I could say some of the species are facing climate impacts, climate caused extinctions, as in the case of several other species. Bivalves are most diverse. It's got over 11,000 species. India has got 11,000 species, followed by cephalopods, about 210 species. Gastropods have about 190 species. Polyplacophores of almost 41 species. Scaphopods almost 20 species. If you look at the exploited resources of molluscan, you can see about 150,000 tons in cephalopods, over 100,000 tons in bivalves, about 20,000 tons of gastropods are exploited from Indian waters. I've just given you a graphical representation on the right side. You can see the molluscan production for the global one, which I've described on the right side, and the production from overall group you have given here, the FAO source. If you look at the neritic squids, Euroteuthis, Duaseli, and the pharaoh cuttlefish, the Sepia, Pharaonis, or the needle cuttlefish Sepia aculeata, together contribute about 84% of the total cephalopod production in India. Among the west coast of India, you can say that the Euroteuthis Duaseli contributes over 50% of the landings in India, followed by the Sepia, the two species of Sepia, which is contributing about 47%, that is Pharaonis and the aculeata. Among the squids, Euroteuthis species, and among the cuttlefish, the Sepia form the significant part of the contribution of the cats from Goa and Kerala coast, respectively. Number of octopus species, namely Amphiooctopus membranaceus, forms about 1% of the catch, mainly from Kerala. The dominant species of landings along the east coast is basically Sepia Pharaonis, followed by Euroteuthis Duaseli and Sepia aculeata. In bivalve mollusk, you see the clams, cockles forms a significant contribution. If you look at Bilorata, Cyprinoids, Merytrixcasta, Marcia, and Catalacia form a significant contribution, about 73%, 73.8%, followed by oysters about 12.5% or 13%, mussels about 7.5%, and if you look at the window pane oysters contributing about 6.2%. So I've just shown you the production graphical representation on the right side. And for your information, you can see this is the source of CMFRI, Central Marine Fishery Resource. Of course, we know that marine mollusks are indicators. The great Goldberg and other scientists, long back in 1950s and 70s, postulated the mussel watch program, where they function as important indicators of climate change, maybe in pollution, maybe on temperature, maybe on salinity tolerance. All these are very wonderful creatures. Macrobenthic invertebrates are also considered important indicators of aquatic diversity, aquatic ecosystems in diversity, variety of environment stresses. Mollusks have been major calcareous organisms with extensive fossil record. They can provide important information on past climatic events and ocean changes. And if you look at the oceanic changes over the paleoclimatic scenario, you could see that sclerochronology, which you will be having exclusive lecture later on, it provides periodicities retained in the accreted hard parts of the bones and diverse living and extinct creatures known as sclerochronology, which will be, and the growth rates on this and the aging and over the period of century and the growth rates will provide you the actual environmental constructions. And one of the important aspects of this is the Cornell paleontologist J.W. Wells is one of the most exciting discoveries of sclerochronology in 1963. He discovered the current corals create 365 days growth increments in their skeletons per year. And whereas the ancient corals of the Devonian period have about 400 per year, implying that the Devonian day was around 2 hours shorter than today. The increased number of daily growth increments in fossils, that is the increments of the growth rings, or you can see the specimen here on the cross section on the right side. I think it will be dealt in detail. But what I just want to imply through this to this slide or this information here is the increased number of daily growth increments in the fossils appear to confirm that geophysics has seen, namely that the Earth's rotational phase is steadily slowing throughout the geological time. And the information on sclerochronology help us to understand our cultural complexity found over the archaeological period. And the information on isotopic oxygen and information, it is 18, and isotopic carbon values reflect the environmental conditions, including the temperature and salinity. So the climatic records of the shells are used for interpretation of modern shell condition, ocean conditions in the sites uninhabited by human record keepers, or tracing the seasonal differences in ancient oceans, almost back through the geological time. But if you look at the fossil record also, mollusks have a long Cambrian extensive records. Because of the formation of the gastropod, that is bivalve, protoconch is correlated to the larval life. Example, the lithotrophic versus the planktotrophic. Fossil record provides an information on the life cycle, and you have one of the largest species of mollusk on the fossil here. So all these, and another ammonite on the heteromorph undescribed. So all these species of mollusk, which are currently the Lopho Trochozoa and the true Trochophore larvae, it's clear that they are not eucoelomates, that are serial repetitions of organs, are independent of the Annelida, that this direct sister groups remains debatable. So the progress, what I just want to stress here, that because of the climate changes and the climate, because of the carbon dioxide release, because I highlighted that carbon dioxide has extensively increased or risen since last 150 years, risen by 36%. And if you look at the most recent 2014 IPCC report, the carbon dioxide has almost reached 370 or to almost 410 ppm. And it's also proposed that, or it is postulated that in 2070, the carbon dioxide would be rising to about 560, it's estimated. Even the pH is very, very important for the existence of these important molluscan resources, especially the larval forms. If you look at the larval forms, it's very, very important. And the larval forms are very much, and the pH of the day, as per the IPCC report, if you look, has decreased from, it is almost zero point 0.1 unit of that, almost 30% rise in pH. And this is from 1800. 1800, 16% decrease in carbonate ion in the ocean waters. And if you see that mollusks are good proxies for paleo, environmental, and climate studies. And if you look at the 2014 IPCC report, it is highlighted that pH could decrease further to 0.14 to 0.35 units. So what happens with this such a change? What is happening? So if you look at the phylogeny of these species, also the changing phylogeny of the species, I've just shown you a phylogeny of a species most recent, and which shows that there's all these recent tree of Aquilifera and different species of the monotype Monoplacophora represents a highly specialized groups of the, whereas there is a progress in our understanding of the Codofoviate evolution. A phylogenetic system of Selenogastrus is still infancy. In contrast to last 20 years witness substantial progress concerning the internal phylogeny of Polyplectophora, Bivalvia, Gastropoda, and Cephalopoda. So this change over the Silurian period, Ordovician, Cambrian, and Precambrian period, almost up to 650 million years ago. The changes over the years and over the phylogenetic modifications have seriously, and this would be one of the climatic factors which is seriously influencing. Considering numerous aspects in which molluscan developmental biology play an important role. As I highlighted, pest control, protein production to evolutionary oriented research is important. But what we understand is that if you look at this, the different annelids, or if you look at the different molluscan species which have radiated over the period, you can see that the potential of, however, has been scarcely exploited, to which at least partly due to the lack of solid code of model system species for various class level taxa in which ontogenic material has been readily obtained. But the 5 main taxa, which is usually referred to as the classes, are the two worm-like, that is the Aplatophoran, that is shell-less groups classes, or the Selenogastrus, or the Neomeniomorpha, or the Ventropliceta, that is less than 300 species, or the Codofoviata, and the Chitodermomorpha, that is about 150 species. And also the chitons are there, which is about 100 or about 1,000 species. Tusk shells are another group of species, which is about 900 species. The limpet type species, almost a class belonging to Monoplacophora, that is about 30 species. The size of extant species of mollusks, that is ranges from 0.4 millimeters to 7 millimeters, as in the case of Achiteuthis, that is body without arms of a giant squids. So I have just shown the giant and colossal squids almost. So mollusk, if you say as a species or a group of phylum, it's large. It's extremely large. I said it's over 100,000 species. No other phylum with the animals have so many species under threat of extinction. Life histories, if you take, they have different life histories. I don't want to elaborate, but for information, life cycle starts with spiral quadrate cleavage to prostomy, embryonic development from planktic to originally lithotrophic to trochophore larvae, which is equipped with prototroch. You have seen the different molluscan stages out here, and the trochophore larvae. Benthic adult has also got metamorphosis to several Selenogastrus to protobranch bivalves to Pericalima larvae. Gastropods and bivalves have been independently evolved. Planktotrophic veliger larvae show intracapsular or direct development. Several partly parasital larval species, that the Glochidium, are found in the unionoid freshwater bivalves. Cephalopod producing very large and yolky eggs causes the superficial cleavage similar to that of reptiles or the birds, and no sign of trochophore or veliger larvae in the case of cephal. Habits of, if you look at the habitats, mollusc classes have restricted marine habitats. As I said, it represents freshwater, brackish, and most successful colonizers of the animal kingdom. And they are dominating, are rare. That is example in the case of pteropods in cold waters. terrestrial species extremely restricted or distribution or single species in valleys. Consequently, if you look at the species of Indospenia caelum, it's a species restricted to Kerala backwaters. This another species is also restricted to Kerala backwaters, the Kerala coastal backwaters, which you see the Nosodonta insectness. There's also habits and lifestyles. If you look habits and life cycle, if you look, they may be burrowing their head and softly, very sure. They may depend on the different sizes, well-known swimmers, heteropod, cenogastropods, certain ophistobranchs and chiropods, or Spanish dancer and sea hare, the aplasia, and the squids. The squids, you can see different types. Some may be semi-sessile mollusks. All types are having a feeding of mollusks. They may be predators, they may be detritivores, they may be filter feeders and parasites. You can see a Xylophaga nandani. This is a species which has been recently named after some of my Dr. Jayachandran, my student after me. This is a deep sea marine wood boring mollusk. It's a wood boring bivalve mollusk, belong to the phylum Xylophaginidae in the Eastern Arabian Sea. This was recently published in Marine Biodiversity. And several gastropod bivalve taxa housed in the symbiotic bacteria of zooxanthellae become the least chemo or phototropic and use biochemically inert food and nourishment. So what we see in man and mollusks are significantly related. But when I say about climate, the climate is something which is very, very significant, because if you look at man and they have significant cultures across. They have socio-economic, agriculture, and many are become animal pests. Oysters, scallops, cockles, mussels, octopods, cuttlefish are all good for protein source. Many octopod and scaphopod shells are widely decorative and monetary purposes. Attractive conch shells are they're also fatal if their neurotoxins are got onto the body of animal or humans and they have conch shells produced by the salivary glands for pain relief and treatment of chronic neural diseases, including Alzheimer's and Parkinson and depression. The current approaches to the continuation of long-term tradition of molluscan contributors to neurobiological research is also significant. Now, if you look into their cultural role, they have a very important role in food, tourism, curio, different ecology, pearl production, and also food and security. They have important biosecuritors, bioindicators. But what I would significantly say here is that mollusks they deposit large amount of calcite or high amount of calcium, magnesium, and calcite in their skeletons. And most of the larvae have aragonite in the shell, so very vulnerable to increasing climate, temperature, and acidification. That is well. And as I highlighted earlier, the ocean acidification is significant here. And the ocean acidification, because of your increasing carbon dioxide, will increase, and ocean acidification will decrease in the production and accumulation of marine biogenic carbonates, less favorable for the biogenic calcifications, and it causes severe impacts on marine calcifiers and marine biogenic processes in the ecosystem. So this kind of impact, because of the climate, because of the planetary boundaries which we create because of our own human actions in the Anthropocene period, is significant. The ecological perspectives on mollusks recognized as bioindicators because of the environmental change and ecosystem, as well as in several ecosystem services. Of course, they are very significant in cycling and storage of carbon and nutrients, water purification, stabilization of shorelines, habitat structure, and food source of organisms. They're very significant in habitats and via production of shells, and affect abiotic conditions and resources available to organisms, and persist for centuries or more, producing long-lasting ecological and historical legacies. They are notorious pests, invasive and pests, because some of the, including, they may be parasitic parasites, including flukes and nematodes. As I already informed earlier in part of this lecture, they may be intermediate host and cause severe medical problems. Some of the pests which we have described from our own lab in Cochin University of Science and Technology is a semi-sessile bivalves of charu mussel, the Mytila stregata, black strip mussel, the Mytilopsis salai, and the zebra mussel, Dreissena polymorpha, or inflict economic damage as biofouling organisms attached to the plumbing and ship bodies of the bices threads. These are all reported from our group of researchers. They're highly broad and Neosian mollusks, particularly broad ecological tolerance that resulted in rapid colonization or formerly non-native species in different parts because of the consequences and replacing the native fauna. High efficiency of these species, because Phycilla acuta is another species which we reported very recently. It's an invasive species of aquatic pulmonate gastropod Phycilla acuta that has spread out the growth last century, and is also which is also reported from one of the canals here, and large number of canals, and causing severe ecological and health problems to the public, and also causes severe medical issues. So the threat to mollusk is also serious. But when I come to the threat to mollusk, we should see that the molluscan resources, if you look at the coast of the Mediterranean, recent information suggests that climate is warming or changing along the eastern, if you look globally at the eastern Mediterranean also. Native flora and fauna are facing rate of extinction. And recent report along the in the Suez Canal and the Israeli coast, and reports by University of Vienna by one of the researchers in their University of Vienna by Albano, reported that 95% of the molluscan shells are found, and they are only shells are found almost by scuba diving in the deep Mediterranean. Almost many are disappearing. And they are being, and if you seriously look into the aragonite formation, the ability for calcite, the mineralization, the calcite formation, especially in the young ones, the molluscan young ones, the veliger larvae, the trochophore larvae, the molluscan biomineralization pathway has changed. Molluscan biomineralization and calcium carbonate in the form of calcite and aragonite, and many species add magnesium calcite in different pulses. So most aquatic species, as we know, build shells in isotopic equilibrium, and surrounding waters and species use oxygen, we know, in ingested water and carbon from both respiratory carbon dioxide and food source from their shells. As we know, magnesium and strontium may not be the equilibrium with the environmental isotopic content in many molluscan species. So the changing carbon dioxide, the changing ocean acidification. Ocean acidification is significant with regard. Our studies on Antarctica, the Arctic Islandica in 1767 is one of the very important proxies where a lot of studies have been done in the North Atlantic and the Arctic climate change. That's why I said when you link to molluscan population to severe sense of habitat loss, overexploitation, marine pollution, and also because I said that marine mussel watch was a significant program on invasive alien species, climate change. If you look at the Gulf of Mannar in India, pearl oyster fishery has collapsed over the last few decades in the 20th century. Climate-driven issues of ocean acidification are significant here. And if you look at the glossus species, another good proxy where changes have been reported from the Arctic areas and the Mediterranean. Shell geochemistry, that is also very important in studying these climate change affecting the molluscan species. Stable isotope and trace element profile, especially uranium, reflect the isotope chemistry and composition, providing information on hourly, daily, millennial, and the past and centennial condition of the mollusk you can get by using as I highlighted above, the oxygen 18 and 14 carbon isotope ratio provide to reconstruct even in estuarine habitats. And also track with the river inputs, whether the source of contamination, what are the sources of it. Of course, we all know that the AMOC, Atlantic Meridional Overturning Circulation, which is happening throughout, it has changed over the 17th century in the Ice Age, has transformed the Arctic species, Arctica islandica. Similarly, in Mytilus species has been widely studied globally, especially with relation to climate, ocean acidification, and warming with Mytilus edulis. Their shell morphology, the shell mineralogy, shell calcification are all being studied largely. And survival rate, not affected by high carbon dioxide. And they found that the adult survival rate is not seriously affected by high carbon dioxide and led to mortality with acute pH values. See, we know that the calcareous organisms especially show lower growth rates, thinner shells, and changes in protein expression when compared to normal species. So one of the greatest influences of climate change, greatest changes in the warming, we know the rate of warming is almost 0.83 degree centigrade, and almost the climate temperature globally is rising by almost 1 to 2 degrees centigrade. It can vary in different ages, especially in the coastal areas. And we can see species like calcareous organisms. These are calcareous organisms with having aragonite, which is having calcite, biomineral compounds, and the shell thickness, the thinning of the shells, and the change in the protein expression is also very significant. And more investigation and the geochemistry profiles have to be taken to study these. Arachis misera from the highly acidic areas of the Taiwan coast had thinner and more globular shells than those of the less acidic areas. This is because has been studied and reported from the Taiwan area. Even several abalone species, several studies have reported the smaller larval shells and larger and longer early development under acute pH levels have been reported. Even swimming activity of the larvae and dispersal capability of Nassarius species larvae negatively affected high temperature with high mortality and leaves and less recruitment rates, impacting juveniles and adults. So ultimately, the synergistic effects of acidification and temperature warming, also other than Mitulus, in Litorina litoria, in the temperate region show decrease in the respiration rate. So there are also several biomarkers. Also, one of the studies by Hebanthal in 2013 reporting increase in stress biomarkers, lipofuscin in Arctica, Islandica grown under low and pH and high temperature. Mitulus edulis have been studied for the last 50 years in several models. That's what I would like to say. Different models. And say that the decreased, even if you look at the US East Coast, it decreased over the East Coast, and the species of Mitulus trocylus was replaced in the West Coast. And the species of Mitulus trocylus was replaced by the invasive Mitulus galloprovincialis in salinity-mediated zones. So such kind of species transitions, species changes, invasive species have all affected the molluscan species along with the other large species changes, or I would also say like species modifications have been taken, including the molluscan resources. Recent studies show that the marine mollusks suffer the highest extinction rate. Government of India has reported almost 24 species of marine mollusks in the various schedules of the Wildlife Protection Act. I would say that with this, I would like to say, I would like to conclude I think I have almost taken 15 minutes additionally to the time allotted to me, because this is a very interesting area. I would say that, and it's very interesting because I have just almost dealt almost 50% or 60% of it, because areas of relationship with agarite production, a lot of studies are globally. And my friend Dr. Alan will be giving you the next lecture, I understand, will be also chronology. It's a very important area associated with. I have just spoken a little bit on that. Thank you all for a very patient hearing, and thank you all with a very good evening from India. Thank you all, friends. I think it has been useful. I think. Thank you so much.
Thank you, sir. We sincerely thank you for taking out time from your busy schedule to provide us with such valuable information. Now we have the time for an open discussion. Participants, you can ask your questions in the chat box now.
Yes, I have few. I have the first question. Why? Yeah. I'll be answering. are all theropods. Yeah, this is a very. I think I have. Who you will be reading that question? I'll come to the first.
Needu.
Okay, thank you. Yeah, I have first question, sir. What is the reason for the fossil mollusk like ammonite or the shalligram geographically isolated and observed in river Gadaki, in Nepal? Yeah, that's a very interesting question, because I basically I'm not a deeper molluscan scientist, but still from my knowledge, it should be because it is because of the geological timing. I think so. It is geographically isolated because the fossil deposits, during these ammonite and shaligram deposits are maybe because of the fossil geological time scale would be the reason before these differences in the observed in these areas. That should be the reason. I'm not very. Dr. Hitesh is there on this.
Yes, sir. Good evening, sir.
Good evening. Good evening. Yeah, basically it is more related to the paleogeology or paleology mostly. You have any idea on this? My thinking is it should be because if you do a fossil study, if you do an isotopic dating, you will be able to know the ammonite. Because when I was surprised to see ammonites when I visited the New Zealand, many years back I visited the fossil beaches of New Zealand. I was surprised to see ammonite. Just if you break rocks, you can see so many ammonites in the open in the coastal beaches. It's very surprising. So they were doing so many kinds of isotopic measurements, and the reasons they were highlighting it is over the changes in the river inputs, so many kinds of salinity variation, the geochemistry would all be reasons, and of course they are geographically isolated, definitely. And specifically who are working in paleogeology, paleoclimatology will be more strong in answering that question. I think I'm there in that. Okay, Dr. Jitesh, fine. Can you hear me, Dr. Hitesh?
Yes, sir. Thank you very much.
Thank you.
It was really a very informative and interesting talk, sir.
Yeah.
I myself is a marine zoologist, so this was a very keen and interesting area that I really loved listening to, sir. Thank you so much.
Thank you. Why cephalopods are also known as advanced species, and yes, cephalopods because by their phylum and their zoology, their changes in the diversity, because their developmental scenarios, the developmental models are very much related, and they are far ahead. If you took the squids, the octopus, and so many kinds, they are far ahead in their perception, even in their growth, in their even that cephalopods communicate. They will communicate. Squids communicate. Giant squids. So that should be, and they are definitely far ahead in their perception. The genetic variability, the genetic enhancement is also very crucial. Thank you. Then there's another question. Yes, theropod has good indicators. Not much studies have. See, why I say these butterflies, the ocean butterflies, because they're very indicators. When I was in the Arctic, very interesting it was. We could see these theropods, and they were good indicators of metal contamination. There were a lot of mercury contamination in the Arctic, the north, the poles, when we were many years back. So there were theropods, the sea butterflies, and they were very interesting, and they are good indicators. But mind you, friends, a lot of work has been done in the as I highlighted on Arctica species. Then I said about the Mitula sedula species. Then also I told about the Anarchis species. Then also I spoke about the Glossus species. So if you look at the theropods, when you scan through literature, we find and look at the information. And this is one of the very interesting area where people could take up, researchers could take up on this area, because you have isotopic ratios, and other than the isotopic ratio and the changes and from the oxygen and carbon ratios, and also the sclerochronology, infrared thermography is also one very important, where you can, this is what I just missed in my lecture. Infrared thermography is one important area where researchers can start up, uh, because why I said is, there you can look into the population, the ocean acidification linked to the pH and temperature, and also salinity. These could be important, and infrared thermography can provide some information on really real minute and subtle changes in the, especially in the larval forms, the developmental stages is very interesting. And theropod are good indicators. Definitely, we have found the metal contamination in the Arctic, but seriously telling, there are very not much studies on theropods when you go through it. What will be the future of molluscan diversity in the next 20 years? It's very appalling, I think so. Very appalling. Will it be affected? Certainly. Yes, it's going to affect tourism. When we look into the Indian scenario, that's what I said. Because I have just shown you several species of uh, molluscan resources which have been there. There's a shift in the native species. Highlighted that I also told about the species of along the U.S. East and West Coast. There's a species which is transforming and it's taking over Mytilus edulis by Bitinus, another species of Mytilus, and which is also something like a scavenging species. Here also we found Mytilus salai and other invasive species we have found in the Indian subcontinent. So these invasive species, as is regular under standard marine biology we study, they are all invasive species. They have shorter food chains. They have opportunistic species. They're enhancing and taking over the niche and the ecosystem and the environment of the native species, and reducing its food, and even in its developmental, and even the developmental stages of the native species are disrupted. So this is very the larger effect of climate change, and definitely I would say theropod and other, and this is the molluscan diversity. Kevin, is going to be grim. I would certainly anticipate, because let me tell you, friends, I have been a researcher who has been working on the benthic diversity for long. We have been working on different invertebrates from the Indian coast, from the Arctic coast, and of course in the Indian Ocean, different coastal zone of the Indian marginal marine ecosystems, and also in the Arabian Sea. There we find that the diversity is seriously depleting, and if you look at the even in the Arctic biodiversity, Arctic we say it's pristine. There also the diversity is sincerely not encouraging. So all this shows that the calcification issues, the ocean acidification issues are looming large on related to the climate science or the climate change. And 2014, I said, that's what I said. There's a warning which has come in the very recent 2014, and also the very recent 2021, which is also repeated. I think so. The ocean acidification and pH is going to be still it's going to further decrease from 0.14 to 0.35 units. And still more is postulated. Can weak acids like carbonic acid also? Yes, certainly. Yes, certainly. Yes, acidification is certainly, it's going to be a serious issue. And I'm so happy. I think which species we use as climate change analysis of, particularly gastropod. Oh, which species we can use as climate change study, particularly the gastropod. I have largely worked on the bivalves, but there are several species of gastropods which can be used, and mostly that I can give you, because there are whatever I looked as which I've seriously studied deeper was in the bivalvian species, the Metulus, the abelons, And there are other several very interesting, but not much work has been done on the gas reports. But gas report is also a very significant indicator of climate change and the studies which is there. I think I've answered to mostly to your questions. If you have still questions, you can email to me. I'm sending my email ID also. I think Vishnu can. Uh, uh, I've just yes, I can send. I
Okay,
think
great.
Vishnu can email it. Uh, Thank you all, and thank you so much, and thank you all. I hope I have done justice to the lecture today. Thank you, and all the best to Dr. Alan also for his next lecture. Thank you all, dear friends.
Thank you, sir. Thank you.
Thank you, sir. I hope all participants had a very valuable time in this session. Once again, thank you, Bijoy, sir.
Thank you.
While moving on to the next session, here we have very renowned personality Dr. Alan D. Wanamaker, Professor Iowa State University, USA, who is going to make familiar with the very recent topic, Sclerochronology, a window to paleoceanography. I
Technology.
repeat the topic, Sclerochronology, a window
Technology.
to paleoceanography. Dr. Alan D. Wanamaker, the Department of Geological and Atmospheric Sciences, is a trained geoscientist who is interested in using geochemical and sclerochronological techniques to document and better understand changes in Earth's climate and ecosystems through time. He has 156 publications. His research interest is developing new geochemical tools and proxy records for paleoclimatic applications. He has done four major projects on the topics: Portugal hydroclimate, unexpected isotopic variability in biogenic karaganite, a user issue or proxy problem, Proboscidean population dynamics in the mid-continent, etc. Welcome, sir. It's over to you.
Thank you. Can you hear me?
Yes, sir. Uh,
Yes, sir.
yes.
Yes,
Yes,
sir.
yes. Good. Please.
Okay, I'm having trouble sharing my screen, so I might have to restart real quick here. Just a moment.
Okay, sir. Okay. Uh,
Okay, just one second.
yeah. Yep. He will come back, and he will come back soon.
Oh, still having trouble sharing my screen.
Uh, uh, mm, mm,
There we go. Sorry about that.
Okay, sir.
Can you all see the screen now?
Yes, sir.
Okay, so sorry about that. There's too many different programs we can use. So yeah, good morning from Iowa and the United States. And I really want to say thank you for the invitation to present to the international webinar with a focus on sclerochronology and oceans and climate. So what I'll do today is present some work that our group has been working on for many years now, looking at 3 distinct oceanographic areas over here in the northwestern Atlantic, the Gulf of Maine region, the North Icelandic Shelf, and over here in northern Norway. And the theme of the talk is looking at variability in the North Atlantic Ocean to try to determine what has happened in recent centuries to 1,000 years, and then better understand what is happening in the modern period. So first of all, I would like to acknowledge my contributors. And there are a lot of them. Those are shown here for the Gulf of Maine work, many students, Norway also students, and my colleagues from Iceland, and the work has been generously supported by the National Science Foundation and the European Union to look at climate over the last 1,000 years in the European area. Okay, so I like this slide a lot because it gives you an idea of what we know about the recent past, about over the last 1,000 years. And you can see the relatively short instrumental period here and some projections forward. So the point is that the instrumental record is too short to constrain the variability of the climate system, which changes on millennial timescales, centennial timescales, decadal, annual, seasonal, etc. And by having good quality paleo data, we can better constrain what is happening in the modern period, which will allow us to project what might happen in the future. And these projections, of course, are largely based on the amount of CO2 that we continue to put into the atmosphere. But a little more context. So using paleo data in modern climate studies, the paleoclimate data can help place modern oceanographic change into context. Paleoclimate data can expand our temporal understanding of natural climate variability in the global ocean. And paleoclimate data coupled and merged with instrumental data will facilitate hypothesis testing regarding the mechanisms that influence regional ocean climate variability. And again, the oceanographic areas I'm interested in talking about today are centered in the North Atlantic, and you can see largely that each of these are situating on some type of ocean boundary in virtually every case here. And in particular, the locations of these studies are within the influence of the Atlantic Meridional Overturning Circulation, or AMOC, which is shown here in schematic, with warm water coming from the subtropics flowing northward toward Scandinavia and the UK and into the Arctic. That water eventually cools in the wintertime, becomes dense, and it sinks over here in the western boundary area out of the Nordic seas and flows back southward toward Antarctica. Shown here up in the right panel shows you the global extent of the meridional overturning circulation, but I will focus largely in the Atlantic sector today. So how do we do this? We use these long-lived mollusks. The Arctica icelandica is shown here. These are some of the shells, and these are the internal increments within the shell in the hinge region here, and out on the shell margin over here. And clearly there is information in there, and we happen to know that these are annual based on lots of reasons. So they have prominent annual growth increments determined in the 1980s. They're quite long-lived, and they can live several centuries. And like trees, they can show a synchronous population in terms of their growth. And so if you get a collection of shells in the same location, in the same water depth, the same conditions, they tend to all grow in the same way year to year. And there's been a lot of work to explore the geochemical information stored in the shell aragonite, in particular radiocarbon, 14C, oxygen and carbon isotopes, to better understand the past variability in hydrography in ocean dynamics. So on to a little bit of fun. This is a location here in the Gulf of Maine, and we're working with a local fisherman who fishes for lobster, but occasionally we can hire them and they'll go out and collect some of these quahogs for us, Arctica icelandica shown here. And depending on the location, you can get all sorts of things that come up, but this area is pretty rich with the ocean quahogs here. And these are all largely live caught here, it's shown in this bucket. And so in this particular place, there was probably maybe 100 clams that came up. And at the same time, we'll collect these shells here, which are the fishermen refer to as white shells. And these are just often dead single valves, but from the same exact location. So we can try to add these to the record. So after a bit of fun in the field, back to the lab we go, where we take the shells here and we section them, and we embed them in epoxy, and then we cut the epoxy, and we do a lot of polishing, and then we work with a shell block like this so that we can see those increments. And again, this is an example. I'm sorry?
Excuse me, sir, the slides are not visible to us. They are not moving.
Yeah.
Okay, could you say that again, please?
So the slides weren't visible.
The slides aren't moving?
Not moving.
No, sir, no.
We can't
The screen sharing has stopped.
see your slides.
Okay, we'll try it again. And can you see that now?
Yes, sir, yes. But can you put it in the full screen?
Slideshow.
Is that working?
No.
No, sir.
So you can't see the screen?
Yes, sir.
Yes, sir, but
Okay, sir, fine. You can move it by yourself.
yeah. Yeah, okay, sir.
Can you see this screen?
Yes, sir, the
Hello,
screen
sir.
is visible now.
Hello, sir. It's me, Jishnu here. So I think the slide is visible for us, but whenever you change the slides, it is not visible for us. Like whenever you change we were still in the first slides.
Is that a different slide there?
Yeah, now it is different slides. And can you make the screen more visible? Or can you make that as a slideshow?
Yeah, but when I do that, it seems to not. Okay. Is that a different screen?
Yeah, now we are seeing the preparing for geochemical analysis. That slides.
And then now the chronology?
Sorry, sir?
Okay, I'm changing the slides. Are you seeing that?
No, sir.
Okay.
Yeah, now it is changed.
Okay. And it changed there?
Yes, sir. Yeah.
Okay, so it's just not working in presenter mode. I don't know why.
I think if you change the presented mode, maybe it will work. And if you click on the right side, then it will change. After once you went to the presenting mode, then if you click this right hand slide, then the slide will automatically change.
Did that work?
Oh, no.
See, it's not working in presenter mode. Okay, hold on.
Okay, so then please continue with the same method, no problem.
Is that working now?
No, it's not changed.
No?
No, sir. Sorry. So if you don't mind, can you mail me the slides? Then I will change the slides for you.
Okay. I think it's too big to mail, but I can put it in a Google file real quick.
Okay, sir. Okay.
Okay, I'll do that and then rejoin the meeting.
Okay, sir. Please mail me. Yeah. Okay. Dear participants, sorry for the inconvenience, and Alan sir will join within minutes. Okay, thank you. Please stay on there. Okay, thank you. In a minute.
Were you able to open that file?
Uh,
No, sir, I'm not able to open the file. Please, can you give me the permission to access the file? Unless maybe you may get a mail from me for accessing the document.
Okay, 1 second.
yeah.
Hello, sir. Right now, I forwarded a request to your mail. Can you give me the permission, sir? The mail ID will be shareandgain@gmail.com. That's our webinar series name.
Yeah, did you send that to my Iowa State email? That I'm not seeing it.
Okay, sir.
Okay, sir. Sir, please check your mail now, sir.
Okay. I think it should work now. I did open the other email, the original one. Did that work?
Yes, sir. Thank you so much. I will share the slides with you now. Okay, sir. Sir, is that okay?
It's quite small.
Okay. Yeah, it's too small.
Maybe I can try presenting it from the Google file instead. I don't know if that would be any better.
Yep, sir. Yeah. Because I'm using my laptop for YouTube streaming. Sir, actually, I shared this with my mobile because I'm using my laptop for YouTube streaming. So, yeah. If you do this slide share, then it will be great, sir. Yeah.
All right, let's let me try this. Can you see that?
Yes, sir.
Oh, you can.
Yeah, we can see, but it's small. Yeah, now it's fine. This is visible,
Yeah.
sir.
Does it change?
Yes.
Okay.
Yeah.
Okay, well, again, apologies for the technology issues. I think I'll just, for the sake of clarity, start again. I'm presenting 3 case studies with lots of collaborators from the Gulf of Maine, Norway, and Iceland. And I had mentioned earlier that the reason we want to bring in some paleo data, as is shown here, and can you see my cursor?
Yes, sir. Yes.
Okay, great. The paleo data here really provides some context for the short instrumental record shown here in this dark color. And the paleo data will allow us to better constrain future predictions, and as is shown here in terms of temperature anomalies, really depends on the amount of CO2 that we continue to release. So briefly, the paleoclimate data can help place modern oceanographic change into context. It can expand our temporal understanding of natural climate variability in the global ocean before humans started to have such a large control. And we can use paleoclimate data with instrumental data to facilitate hypothesis testing regarding mechanisms that influence regional ocean climate variability. The 3 locations I'm talking about are shown here. Over on the left, we have the Gulf of Maine. Over here on the right, we have Iceland, and over here, even more to the right, northern Norway. All 3 locations are located within the influence of the Atlantic Meridional Overturning Circulation, or AMOC. And shown here in the right panel, the warm, salty water flows northward up along the eastern US seaboard and starts to cross the Atlantic, where it delivers heat and vapor to the Arctic and the UK and Scandinavia. And this water then cools, becomes more dense and sinks in the Nordic Seas and the Labrador Sea, and flows as a deep western boundary current, where it eventually is pulled back up by winds around Antarctica. This circulation has a lot of impacts on global climate, and the locations that we've been studying are within the influence of that overturning circulation. Again, we're using the Arctica Icelandica bivalves because they have prominent annual growth increments. They can be quite long-lived. They show synchronous population, growth population, and we can build master shell chronologies. and the geochemical information stored within them can be used in modern and paleoceanographic settings to reconstruct environmental conditions. Here we're collecting some clams in the Gulf of Maine. This is what a bucket of those look like. There may be 100 live clams inside of this bucket, and also some dead single valves, or white shells as the fishermen call them, and we use those to extend our records back in time. After a day of fun in the field, we go back to the lab for most of the next year working on this material. Here is just a single valve shown here. We cut a section in the middle of it and embed it in epoxy. And that is a picture showing that embedded shell in epoxy, and it's cut down the middle. We polish it quite a lot, and then we get a nice shell block like this that we can work on. Again, shown here are the prominent annual growth increments in a shell from Norway. This is an acetate peel replica of the surface that we use for imaging. We do not do geochemical